Lena, a teenager with a love for fresh fruit, was in the kitchen slicing an apple. She was thinking about her upcoming biology test and how amazing the human body was at healing itself. Just as she thought about how cells divide, her knife slipped, and she accidentally cut her finger. "Ouch!" she exclaimed, quickly grabbing a towel to stop the bleeding. Little did she know, a fascinating adventure was beginning inside her body, right within the dermis layer of her skin. Deep within the dermis of the skin, a quiet but critical part of the human body, the recent cut had set off a flurry of activity. The skin needed to repair itself, and for that, new cells had to be made. This meant that DNA within these cells needed to replicate exactly, ensuring each new cell had the right genetic information. Inside this busy environment, 6 dedicated enzymes and proteins prepared for their most crucial task yet, replicating DNA so that new skin cells could be formed to heal the cut. Helicase, the energetic and curious scientist, was the first to arrive at the site of the wound. She eagerly awaited the moment she could dive into the double helix and unravel its secrets. "It's time to unzip this DNA," she exclaimed, putting on her gloves and turning on the molecular machinery. With precision and speed, Helicase began unwinding the DNA strands, breaking the hydrogen bonds between the base pairs to create the replication fork. As the DNA strands separated, the tension began to build. Enter topoisomerase, the wise and calming doctor. "Hold on, Helicase," he said in a soothing voice. "Let me relieve some of that tension." Topoisomerase carefully made small cuts in the DNA strands to prevent them from coiling too tightly. "There, that should do it," he said with a reassuring smile. As the DNA strands separated, single-stranded binding proteins, SSBs, the diligent protectors, quickly stepped in. "We'll keep these strands stable and protected," they said, attaching themselves to the single DNA strands. They made sure the parent strands stayed apart, preventing them from snapping back together. Next up was Primus, the meticulous and detail-oriented engineer. She approached the replication fork with her tools in hand. "I'll lay down the RNA primers," she declared, carefully adding short RNA sequences to the now separated parent strands. These primers served as the starting points for the next stage of replication. "Remember, DNA polymerase can only add nucleotides to the 3 end of a strand," she explained as she worked. With the primers in place, DNA polymerase, the diligent and hardworking builder, stepped in. "Time to extend these strands," he said, focusing intently on his task. DNA polymerase began adding complementary nucleotides to the 3 end of the RNA primers on the parent strands, creating new daughter strands. He matched adenine, A, with thymine, T, and cytosine, C, with guanine, G, working in the 5-3 direction. "Let's identify the leading and lagging strands," he continued. The leading strand is the one that is synthesized continuously in the 5-3 direction, following helicase as it unwinds the DNA. DNA polymerase works steadily on the leading strand, adding nucleotides smoothly and continuously, with each adenine on the parent strand pairing with thymine on the new strand, and each cytosine pairing with guanine. "Now, the lagging strand is a bit more tricky," DNA polymerase said. "It runs in the opposite direction, so I have to synthesize it in short segments called Okazaki fragments." DNA polymerase carefully added nucleotides to the lagging strand in the 5-3 direction, but since the overall direction was opposite to helicase's movement, he had to work in segments, ensuring that adenine paired with thymine and cytosine paired with guanine in each fragment. As DNA polymerase worked, he was careful to maintain the antiparallel nature of the DNA strands. One strand grew in the 5-3 direction, while the other grew in the opposite direction. The process of replication was semi-conservative, meaning that each new DNA molecule had one parent strand and one newly made daughter strand. Finally, DNA ligase, the patient and persistent craftsman, took his place. "I'll seal the nicks," he said calmly. DNA ligase joined the Okazaki fragments on the lagging strand, creating a continuous and complete DNA molecule. "My job is to make sure the new DNA strands are perfectly intact," he said as he worked. As the enzymes and proteins completed their work, they could see new skin cells forming, ready to repair the cut. Their teamwork and dedication had once again ensured the continuity of life. Within the dermis of the skin, the replication of DNA was a crucial step in the healing process. The enzymes and proteins knew that their work was far from over, but for now, they took a moment to celebrate their achievement. And so, deep within the dermis of the skin, The story of DNA replication was written anew, a testament to the wonders of biology and the unyielding spirit of scientific exploration. The cut would heal, thanks to the diligent work of helicase, topoisomerase, single-stranded binding proteins, primus, DNA polymerase, and DNA ligase.